Hello students, this video lesson is for first year BSc first semester, subject Food Science and Nutrition, paper Human Physiology. This is the continuation of digestive system for digestion and absorption of foods. Digestion of major foodstuffs is an orderly process involving the action of a large number of digestive enzymes. The action of enzymes is helped by the HCl secreted by the stomach and the bile secreted by the liver. Absorption refers to the movement of nutrients, water, and electrolytes from the lumen of the gut into the cell, then into the blood by various transport processes. Substances pass from the lumen to the ECF and then to the lymph and blood, similar to those that are responsible for transport of substances across the cell membrane. Now we have observed or studied the different structures and organization of digestive system, and in this system how the food ingested is digested and absorbed. Now we will learn. This picture shows the intake of food through the mouth and how it travels through the digestive tract till the defecation. The food and drink intake from 2000 ml added with the digestive secretions, gastric secretions, liver secretions, pancreatic and intestinal secretions, they make up about 9000 ml, and about 7800 ml of ingested liquid along with the solid foods is absorbed in the small intestine, and the remaining amount moves to the large intestine where about 200 ml of colonic secretion occurs. Colon absorbs from these 400 ml of liquid chyme about 1250 ml is reabsorbed back in the large intestine. Only about 150 ml of liquid feces is excreted from the body. Now, let us study the digestion and absorption of the nutrients one by one. Let us now study the carbohydrates digestion and absorption. The daily intake of carbohydrates represent approximately 50 to 60% of the diet. The principal carbohydrate of food are polysaccharides, disaccharides, and monosaccharides. The polysaccharides being starch, cellulose, and pectin, and these cannot be digested by the enzymes in the human gut. Glycogen, here glucose molecules are mostly in long chain. The disaccharides being sucrose, lactose, and maltose. The monosaccharides like hexoses, like glucose, fructose, galactose, etc. Pentoses, they do not occur in free form but found in nucleic acids and in certain polysaccharides, for example, pentosanes of fruits and gums. The digestion of carbohydrates starts in the mouth by ptyalin or salivary amylase. producing alpha-limiting dextrins and maltoses. In the stomach, the HCl of the gastric juice hydrolyzes some sucrose. In the duodenum, alpha amylase of pancreatic juice rapidly converts all form of starch and dextrins completely into maltose. It acts in an alkaline medium and its digestive activity is increased by the presence of bile salts. Here, the starch and dextrin in presence of pancreatic alpha amylase gets converted to maltose completely. In the small intestine, succus entericus contains disaccharidases, that is enzymes for splitting of disaccharides into monosaccharides. Here, the sucrose in presence of sucrase at a pH of 5 to 7 gets converted to glucose and fructose. Maltose gets converted to 2 units of glucose. Lactose in presence of lactase gets converted to glucose and galactose. Alpha-limiting dextrins in presence of alpha-limiting dextrinase converts to glucose. Now, after this digestion, the absorption of carbohydrates takes place. Monosaccharides, which are formed either in the gut lumen or in the brush border of the epithelial cells, are absorbed from the jejunum and upper ileum. Negligible absorption also occurs in the stomach and colon. In general, carbohydrates are completely absorbed at 100 centimeter from the duodenum, that is, before the remains of a meal reach the terminal ileum. The process of absorption includes monosaccharide movements across the cell into the blood capillaries by simple diffusion and active transport. In simple diffusion, it occurs when concentration of sugar in the gut exceeds that of the blood. Active transport causes the absorption of sugars against their concentration gradient into the mesenteric blood, which requires energy. Energy required for the process is supplied from the cellular metabolism. Now, let us learn the digestion and absorption of lipids. Fats are classified as simple or neutral fats. These are the triglycerides, which are formed from one molecule of glycerol and 3 molecules of fatty acids. The common known fatty acids are palmitic acid, stearic acid, and oleic acid. Waxes are the esters of fatty acids with long chain monohydric alcohols. The second type is the compound fats. These are complex compounds formed from the fatty acids, glycerol, and various nitrogen containing bases and often containing phosphate groups. They are integral parts of the general cell structure and are present in large amounts in nervous tissues. They are employed in fat transport. The main compound fats are phospholipids, which contain glycerol, 2 molecules of fatty acids, phosphates, and nitrogen base, like lecithins and cephalins. The sphingomyelins, they contain fatty acids, phosphates, choline, and complex base, but no glycerol. Galactolipids, they contain galactose, fatty acids, sphingosins, but no phosphate or glycerol. The third type is the associated fats. They are of 2 main types, First one is those that are obtained from by the hydrolysis of lipids. The examples are glycerol, fatty acids, and soaps. Soaps are the salts of fatty acids and are obtained by hydrolysis of fats in alkali. The second type are those components that are associated with lipids in tissue extract simply because they are dissolved by the fat solvents. For example, steroids, that is the hormones of ovary, testis, adrenal cortex, and cholesterol. The, the other one is the fat soluble vitamins, vitamins soluble in fat and in fat solvents. And how these fats are digested in the gut. In the mouth, no digestion of fat occurs. However, salivary lipase is active in stomach and can digest as much as 30% of dietary triglycerides. In the stomach, gastric lipase, which has a weak fat splitting enzyme, it acts at an optimal pH of 4 to 4.5 because gastric juice pH will be 1 to 2, and it is inactivated at pH 2.5. Fat digestion is also called as lipolysis in stomach occurs only in exceptional circumstances when pancreatic lipase may regurgitate into stomach from the duodenum during achlorohydria. In small intestine, pancreatic lipase and bile salts enter the second part of the duodenum at duodenal papilla and begin the fat digestion. Pancreatic juice is markedly alkaline in nature that converts and adjusts the highly acidic chyme to the chyme at pH slightly above 7.0, which is the optimal pH for lipase action. Bile salts play a role in activating lipase and exert a detergent action by its hydrotropic action, lowers the surface tension that promotes the emulsification of fats, thereby providing a greater surface area for the lipase to digest. Here, emulsification or breaking of many fatty materials takes place. Lipase cannot act on fat droplets, but it acts at the interface between the fat particles and the water and successfully hydrolyzes the 1 and 3 bonds of triglycerides into diglycerides and monoglycerides with release of associated fatty acids. Most of the dietary cholesterol is in the form of cholesterol esters, pancreatic bile salt activated lipase, and cholesterol esterase of succus entericus hydrolyzes these esters in the intestinal lumen into cholesterol. Now, let us see the absorption of fat, how the fats get absorbed into the blood. The pancreas electrolytes, monoglycerides, fatty acids and bile salts interact spontaneously to form polymolecular aggregates called micelles. Micelles are the water-soluble complexes 3 to 10 nanometer in diameter with variable lipid concentrations. They can dissolve hydrophobic compounds in their interior, and such constituents as cholesterol are therefore made water soluble. Thus, micellar formations further stabilizes the lipids and provides a mechanism for their transport into the intestinal cells. Micelles themselves are passively absorbed along their concentration gradient into the luminal brush border, where the bile salts are detached to return to the intestinal lumen, finally to be actively reabsorbed more distally in the ileum. Fat absorption is greatest in the upper part of the small intestine, but appreciable amounts are also absorbed from the ileum. Movements of villi compress the lacteals and villus capillaries. This increases the mobilization of lipids towards the thoracic gut and portal vein respectively. Cholesterol, like short chain fatty acids, is absorbed directly into the lymphatics and reconverted, that is, esterified. Therefore, from the cholesterol esters. Its absorption requires presence of bile, fatty acids, and pancreatic juice. It is mainly absorbed from the distal small intestine. Non-absorbable plant sterols, such as those found in the soya beans, reduce the absorption of cholesterol by combining with cholesterol for esterification with fatty acids. On a moderate fat intake, more than 95% of the ingested fat is absorbed. Only 5 to 6% is excreted in stools. Much of this fecal fat is derived from cellular debris and microorganisms. The process involved in fat absorption is not mature at birth. Therefore, infants' fecal fat content is 10 to 15% of the ingested fat. More than 95% of the ingested fat gets absorbed to the body. Now let us learn the digestion and absorption of proteins. Amino acid linked together in chain by peptide bond are called polypeptides. All proteolytic enzymes, that is, enzymes concerned with the protein digestion, are secreted in the form of inactive precursors, that is, proenzymes, and activated in the gastrointestinal tract. In the mouth, no protein digestion occurs. In stomach, the pepsins hydrolyze the bonds between the aromatic amino acids and a second amino acid. Therefore, the product of peptic digestion are polypeptides of variable sizes. Here, protein gets digested. Pepsins hydrolyze the bonds between aromatic amino acids, and in presence of pepsin with HCl mixture, optimal activity at very low pH, and the pepsin gets inactivated at pH above 5. Pep protein gets digested in the stomach up to proteases and peptones. They are the large polypeptides. Approximately about 10% of ingested protein is broken down completely in the stomach. Caseinogen, in presence of renin and pepsin, becomes soluble casein, and in presence of calcium salt become a coagulum of calcium casein. It can be readily get digested enzymatically. In the duodenum, the proteoses, which are the digested parts from the stomach, and peptones, in presence of trypsin and chymotrypsin, in alkaline medium about pH 8, gets converted to peptides of various chains, that is, small polypeptides and few free amino acids. These enzymes act at interior peptide bonds in the peptide molecules and are called endopeptidases. Then, these polypeptides, in presence of carboxypeptidases in pancreatic juice, gets converted to lower peptides and free amino acids. These enzymes hydrolyze the amino acids at the carboxyl and amino ends of the polypeptide and are called exopeptidases. In small intestine, protein digestion occurs in three places: in the intestinal lumen, the wall or brush border of the intestine, and the cytoplasm of the mucosal cells. This occurs as some peptides are actively transported into the intestinal cells and hydrolyzed by intracellular peptidases. Peptides in presence of succus entericus containing eripsin, that is a set of mixed peptidases which specially hydrolyzes the terminal peptide bonds, gets converted to free amino acids. The nucleoproteins are digested in presence of HCl in the stomach to other proteins and free nucleic acids, that they are RNA and DNA. These RNA and DNA, in presence of ribonuclease and deoxyribonuclease from pancreatic juice in duodenum, gets converted to nucleotides and nucleosides. These nucleotides and nucleosides, in presence of nucleases and nucleotidases in the small intestine, gets converted to pentoses, purine and pyrimidines. So this is about the digestion of proteins in the gut. Now let us learn how these digested proteins or amino acids gets absorbed into the bloodstream. D amino acids are absorbed slowly by passive diffusion only, while L amino acids are actively transported. L amino acids are absorbed more rapidly than D isomers. These L and D forms are the configuration of amino acids. The absorption of both D and L isomers is coupled to a sodium transport like co-transport of glucose and sodium. The transported amino acids accumulate in the mucosal cell and from these cells they enter the intestinal capillaries passively by simple diffusion and facilitated diffusion. Then via portal vein they reach the liver and general circulation. Therefore, after ingestion of a protein meal, there occurs a sharp transient rise in the free amino acid content of the portal blood, which provides the whole body requirements of protein. There are three separate transport systems for amino acid absorption. First one is neutral amino acid carrier system in the brush border that transports neutral amino acids. It is sodium dependent. Second one is the basic carrier amino acid system, also in the brush border, that transport basic amino acid, which is sodium independent. The third one is the separate carrier system in the basolateral membrane of the intestinal cell that transport proline, hydroxyproline and a few other compounds. Absorption of amino acid is rapid in the duodenum and jejunum but slow in ileum. Of the total protein digested, approximately about 50% comes from the ingested food, 25% from the proteins in the digested juices, and 25% from desquamated mucosal cells. Only 2 to 5% of the proteins in the small intestine escapes digestion and absorption. Now we have understood how the major nutrients gets absorbed into the body. Now let us see the water and electrolyte absorption in the gut. Water is always absorbed in the alimentary tract through passive osmosis via a mostly paracellular route between electrolyte tight junctions. Consequently, water absorption is primarily actuated by active transportation or absorption of osmotic electrolytes, especially sodium. Sodium diffuses into or out of intestine passively depending on the concentration gradient. In addition, sodium is actively transported out of lumen into the small intestine and colon by sodium phosphate pumps. In the ileum and jejunum, sodium transport from intestine to blood is facilitated by aldosterone. In the small intestine, active transport of sodium helps in absorption of glucose, amino acids, and other substances. Conversely, the presence of glucose in the intestinal lumen facilitates the absorption of sodium. Movement of potassium across the gut mucosa is mainly due to the passive diffusion down its electrochemical gradient. There is also some secretion of potassium into the intestinal lumen, especially as a component of mucus. Chloride is normally secreted into the lumen of the small intestine by chloride channels that are activated by AMP pathway. Intestinal cells also take up sodium potassium chloride by sodium phosphate chloride co-transporter in their basolateral membranes. In the ileum and the colon, it appears that chloride is actively reabsorbed in a one-for-one exchange for hydrocarbon, thus make the intestinal content more alkaline. Absorption of vitamins and minerals in the gastrointestinal tract. Water-soluble vitamins are absorbed rapidly. Fat-soluble vitamins like A, D, E, and K absorption decreases if fat absorption decreases due to the pancreatic enzyme or bile deficiency. Most vitamins are absorbed in the upper small intestine, but vitamin 12 binds to intrinsic factor, which is secreted by the stomach. And the vitamin B12 intrinsic factor complexes is absorbed in the ileum. 30 to 80% of the ingested calcium is absorbed by active transport, primarily in the upper small intestine, and some of it is also absorbed by passive diffusion. Fact In males, the amount of iron lost from the body is approximately 0.6 to 0.75 milligram per day, whereas in females, loss is approximately more, that is 1.2 to 1.5 milligrams per day due to extra iron lost in blood shed during menstruation. Average iron intake may be approximately 20 to 25 milligrams per day, but the amount absorbed is equally only to the losses, because if iron absorption exceeds its excretion, iron overload will develop. Therefore, amount of iron absorbed is approximately 3 to 6% of the amount ingested. Iron is readily absorbed in ferrous state, but most of dietary iron we consume will be in ferric form. That's why partial gastrectomy and achlorohydria, the conditions are often associated with iron deficiency anemia. This is about the digestion and absorption of food through the gastrointestinal tract. Thank you.